Welcome everybody again to another episode of the ideaXme show, the show that profiles the humans behind the really big ideas that are shaping the world and inspiring future creation, and for all those that like really great stories. I'm Ira Pastor. I'm your health, aging, and longevity ambassador along for this journey. So here we are in 2019. We are in an era of a $7 trillion total healthcare expenditure around the globe, and about $1 trillion of that being spent on pharmaceutical products. As we've been watching the traditional pharma product basket change over the last century, from small organic molecules, vaccines, to larger macromolecules, proteins, antibodies, and now into this era of cell and gene therapy and other therapeutic modalities, the tools that we have seen that are allowing us to access this new generation of discoveries and development has been changing as well. And one set of tools that we've been hearing quite a bit about lately is sort of this triad of artificial intelligence, machine learning, and deep learning, and the respective applications in the drug discovery and drug development process. So in general, artificial intelligence is defined as a computer system that's able to perform tasks that normally require human intelligence. Examples include, but by no means are limited to, perception, speech recognition, translation, and decision-making. Machine learning is a branch of artificial intelligence that involves methods of data analysis to automate analytical model building. And deep learning is a subset of machine learning that teaches computers to basically do what sort of comes naturally to humans, learn by example. In a sort of non-biotech example, in the case of, say, the driverless car, basically teaching a car to recognize a stop sign versus a pedestrian or a lamppost. In a recent show we did with a colleague from Big Pharma, they highlighted that there's something like 150 or more companies involved in applying artificial intelligence now to the drug discovery process. So this is really an area that's heating up quite a bit. So for today's guest, I could think of no one better to come on and talk for us a while about this exciting area of R&D and some of the real-world translational possibilities than Professor Shai Shen-Orr, who is Associate Professor at the Technion, Israel Institute of Technology, and founder and chief scientist of CytoReason. Professor Shen-Orr has a PhD from Harvard University in molecular cell biology and bioinformatics, and he currently runs the Shen-Orr Lab for Systems Immunology and Precision Medicine at Technion, where their core focus is on understanding how immunity is orchestrated in humans and ultimately how to apply this knowledge towards advancing genomic medicine. He works in a variety of areas and experimental and computational approaches to identify biomarkers, to translate this understanding to model organisms in humans, and ultimately understand the complexity of the immune system. Dr. Shen-Orr founded CytoReason back in 2016 based on over a decade of his research, not just at Technion, but also previous work at Stanford. And with the understanding of sort of how the human immune system is at the epicenter of so many pathologies, both directly, whether we're talking about autoimmune disease, allergies, to its use as a biotherapeutic tool in the case of immunotherapies, to sort of all the secondary aspects of inflammation, whether we're talking about connections to cardiovascular disease, neuroinflammation, Alzheimer's, cancer, and in general, sort of the aging space overall, and what we call inflammaging nowadays. Thought this would really make for a fascinating topic. So all that being said, Professor Shen-Orr, thank you so much for joining us today for a little while.
Thank you, Ira. Pleasure to be here, and thank you for the invitation to talk.
Definitely. So we typically start the show off by just giving our guests the floor to sort of learn more about you, how you got interested in science, how you got interested in bioinformatics, biology, and ultimately, your path towards, at this time in 2019, where you're really at the epicenter of a very hot area in biopharma, not just from sort of the drug discovery perspective, but from sort of the whole development process as well.
Sure. So maybe I'll try. I guess I'll go back a few years when I was an undergraduate. I've always been interested in computer science and the idea of artificial intelligence. And I guess when I started, this seemed more like a dream, but basically the idea of making computers assist humans and replacing humans in some of the tasks, particularly the ones that humans are not good in, and there's quite a few of those. While having some things we do great, there are things, particularly putting a huge amount of information together in one holistic picture, is one of those things that humans don't do well. So I actually started this as an undergraduate in Technion, being interested in these areas, kind of doing some research on it, and ended up as a exchange student at UPenn, where I had to get a daytime job as as in a new field called bioinformatics. Who knew? This was '98, so they paid well per hour. So I took the job. And I fell in love with this idea of actually not just doing artificial intelligence and machine learning, but actually doing this in the context of biology, where you do things that are, you essentially continuously discover the world, which to me was fascinating. Up until that age, at that point, I had not really studied biology. I guess I stopped in ninth grade, and suddenly, and so we're doing things that were cool with machine learning and artificial intelligence. You mentioned, getting a car to self-drive. But when you do this in life sciences, you're actually using computers to make discoveries about the world, or the nature of our world, and that caught me on. And I started off just kind of learning this, and back in early 2000 to actually do this in humans and have applications for health seemed like science fiction. So I started small. I started with E. coli. It's a small bacteria. It has a couple of thousand genes, relatively well understood, and starting to build those bigger pictures of networks of how the bacteria actually works, how all these kind of components we understand and have been researching for many years in a reductionist fashion, gene by gene, suddenly putting them all together and trying to understand how the system works and all. That was, I guess, early 2000s. Moved to a child of the genome era, the sequence guy, that started sequencing the genome and so forth. And I moved as a PhD to Harvard. Still doing humans seemed to have been too complex at the time, and so I took a leap of faith and went from a single cell organism, a bacteria, to C. elegans.
Okay.
It has 1,000 cells or so forth, and and that was a fascinating field to actually try and understand development of the worm. But by the time I was finishing up my PhD, this was 2007, I suddenly I saw simultaneously a few things. A, the human genome has been sequenced, and suddenly the amount of data in humans that was very rare until that point started pouring in. And more data means you can overcome difficult complexity and start making sense of things. And so that seemed like an opportunity. And simultaneously, unfortunately, my father was diagnosed with cancer, with lymphoma at the time. And I was working on C. elegans, doing genomics and all sorts of cool bioinformatics stuff. And I was looking what was happening in the hospitals at the same time. What was my father being assessed with? And I was shocked how nothing of what I was doing at that level of sophistication was actually present in the hospitals. And in particularly, with the respect to the immune system, which seems cardinal to health, and understanding it, the diagnosis was of B-cell lymphoma, and yet relatively nothing was understood, nothing was measured. And so that was almost, from a personal perspective, it was frustrating to see this point. I looked around to to see, well, what could be done? And this is essentially the beginning in 2007, I think, was the beginning of a revolution we're now starting to feel, which was that we moved from the immune system being this very complicated thing that we couldn't really decipher, especially not in humans as a system. We could measure it bit by bit, to developing technologies that suddenly allow us to measure the breadth of this. And if you can measure the breadth of the system, you can start actually thinking about understanding it as a system, putting all the pieces of the puzzle together. I went to Stanford for a postdoc, a joint postdoc with Professor Mark Davis and Professor Atul Butte, a well-known immunologist and well-known bioinformatician. And really, we put together this kind of, I think was quite ahead of the time. At the time, we essentially put this human immune monitoring center together, and suddenly they had a single room in which a patient could give a blood sample, and we would measure everything we could on the immune system in many different levels, and start building this picture. So that's kind of how I got going. And immediately, first time you see data on the immune system come out in humans, you start realizing this has applications for healthcare. Right? You can start seeing what is actually happening in this system whose essential role is to monitor your body and protect it from external pathogens and internally from cancer and so forth. So the applications become clear, and so it was always the passion from the one end, the basic science of understanding the immune system and putting all these pieces together, and immediately feeling like there's an immediate, feedback to the taxpayer as an academic on this, that the application are obvious, that there's an immediate translation that one could do. And so in 2016, as you mentioned, I founded CytoReason. It was a company essentially building a machine learning model of the immune system that puts all this data together that we can measure in humans. And we're doing this across many, many different diagnoses, many different indications, bringing in not just the data where we're talking about just in terms of measurements of gene expression of mRNA, or you can measure now that there's just publicly available a couple of millions of samples that you can just download from the internet. There's a huge amount of a treasure trove of data sitting in pharmaceutical companies, and up until now has just been sitting there.
Right.
People aren't doing anything with them. CytoReason can actually process this data and learn from that data summary statistics, that it can improve its models of the immune system. And in the same time, bringing in not just the machine learning of this data, but the artificial intelligence piece of reading all the literature. Every 30 minutes, we publish a new paper on the immune system in this. So that's kind of humbling. And if it's such a big connected system, you want to put all this data together in one machine learning model. So that's essentially kind of where I am today.
No, it's a fascinating, journey.
A crash course on my life.
That's wonderful. And it feeds right into sort of my next question. And I apologize for sort of earlier on, I guess, giving an amateurish description of what artificial intelligence is all about. Could you, and I don't want you to give a lecture today because that's not your responsibility, but in the sort of the general public's overview of artificial intelligence, they have been treated to, I guess, public debates between Bill Gates and Elon Musk and the late Stephen Hawking about sort of all the dangers of AI. And I think maybe in most people's minds, what materializes when you say AI in health is, somebody pushing a button on a computer somewhere and a moiety of some point, like printing out. But it's a lot more sophisticated than that. Could you just walk us through a little bit of this, what I'll call the triad: AI, deep learning, machine learning, and sort of what's involved from a very top line basis, and how these tools are used?
Sure. So I'll start with deep learning, which I think actually is the most common kind of buzzword you hear out, and actually right now has probably the smallest amount of utility, I think, in healthcare. Okay. And that is essentially, these are methodologies that require huge amounts of data. As you said, learn by example, which means that so the data could be things like your genomic sequences or pathology slides and so forth. You got to learn by example. That means that you need to have a lot of annotation, a lot of information that says, what is it? This example that I'm looking at. You give all of this to the computer, and it looks for patterns that match between the annotated information and what it's seeing in the data, and it's seeing a huge amount. And the problem with deep learning is that you don't really, it's a field in which the computer ends up giving an answer, and oftentimes it looks like the answer is correct and performs better than other machines, than other previous algorithms where less data was required. But you really you struggle to understand why the computer gave that answer. As a field in healthcare, if you were thinking, you mentioned the amount of millions and trillions of dollars in the field and how much it costs to develop a drug can go up to $1 billion. This is actually not an easy thing to swallow, that the computer gave you an answer and you are clueless on why the answer was that, because essentially you need to make important decisions and you want to be able to reason over them. So deep learning currently is being used particularly in looking at genetic variants, where Google has actually put in a really nice application on this, and in pathology slides, as I mentioned, where the mutations, it's an extremely difficult field and still suffering from the fact that there's just not enough examples given the complexity of the problem. Machine learning is actually a very broad term to define looking at many different data sets and trying to pick up signals that humans would, I would say, normally struggle to find, just because of there's so many different dimensions. But as deep learning is almost a subfield of it. But if you look within machine learning, much of it is involved about having training data sets and information in which we have much more interpretable features. And if we look at deep learning, if I mention deep learning, is you don't understand why the computer is actually doing what it's, giving you the answer. In other, outside of deep learning and most other fields of machine learning, you have an ability to reconstruct to some extent an understanding of why the answer is as it is, and you can continuously feed more information and improve your ability on this. Artificial intelligence is that one field where I think the computers really, we're trying to get computers to emulate tasks that humans are actually quite good at, such as reading or speech recognition, as you said. And really what we're trying to do there is actually not, the computers are struggling to do it as well as humans, but we as humans are struggling to do it to the same scale that we can get a computer to do it. So I mentioned the paper reading, every 30 minutes comes a paper. Yes, we can read it better than a computer, but we can't spend all day reading those papers.
Sure.
So we, for instance, at CytoReason have put together this natural language processing engine that's fit particularly for the immune system, and it reads all the papers that's published in immunology and tries to build a system level view of everything, that the knowledge of what humans have made it. Does it do it as good as humans right now? No, not yet. We're working to improve, but it does it at a scale that no human could.
That's wonderful. It's wonderful. It's mind-boggling to learn about this in today's stage, but it's truly fascinating, and the possibilities for the applications. Looking at CytoReason now, and obviously this is all non-confidential stuff, but when I look at the website, you're really involved in a broad array of different aspects of the immune system. You're focusing on biomarkers, specifically in inflammatory bowel disease. You're looking at mechanisms of action, the tissue microenvironment, immune responses in the case of cancers, and then what I'll refer to as sort of the interactome of maybe sort of the multitude of cytokines and other immune modulating substances that occur in tissues. Once again, non-confidentially, can you just walk us through a bit of sort of the CytoReason platform and the strategy in these areas?
So as I said, CytoReason tries to bring together those two arms, the big data arm, where all these measurements are coming together. We take this information for many years. So it's been over a decade now that we can measure all the immune system, but the data has been hard to interpret. And so we've built this cell-centered view where we bring the information to like the lists of genes and proteins that we can measure and put them in a context that it's like how we think about biology, cells interacting and so forth. So that's one arm of this. The other arm of this brings in all the knowledge, all the artificial intelligence information, the literature, as I mentioned, because we recognize that as much as the data is great, we humans understand things that are difficult to parse out of the data every time alone. So we bring the two together, and seeing so many different data sets together comes now what it would be the application of such a process. And so when we started the company, we looked at the drug development process and what are the big decisions that pharma needs to make as it develops a drug, right? So the first question comes down to what would be the target to hit, right? And normally comes from how do scientists make decisions? They read a few papers, right? They see somebody publish an interesting result. They make a few kind of intuitive guesses or leaps of faith within after they've read, and they end up having some hypothesis on a target that they then try and do experiments to validate. But given I just described a process where there's actually there's not necessarily any previous data on this on whatever target that is, versus if you look in this day and age, there's actually quite a lot of measurements that could inform this decision making of what would be a good target. So So CytoReason comes and says, okay, well, if we wanted to be choosing a target, why do this without looking at all the previous data? In the 21st century, there's so many millions of data sets of data measurements that we can look at and data sets and so forth. Let's feed in and try and identify targets, prioritize them, and so forth. Goes back to say, I found a target. What would be the right disease in which to hit this target, right? Where does it play a role? Could I prioritize, could I prioritize where would be the first disease that I would want to be developing a drug program in? What would be the second, say it succeeded, how would I expand to other diseases? What would be the second, program for this drug and so forth? Mm-hmm. So we move from looking just at the target to looking at the disease context and looking at the combination of expanding, an indication expansion, if you like.
Sure.
And finally, comes this whole field of biomarkers, which really drills down to the fact that for the majority of drugs, you're talking about only, depending on the drug, but could easily be only 40% of the population responding or less.
Right.
In cancer, it's way less than this.
Yeah.
And so you want to be identifying what are the people who are going to be responding to this drug. It's going to strongly affect your clinical trial success. So you may want to be knowing this ahead of time. And in fact, you may be wanting to identify subsets of the disease, because
Sure.
different subsets of the disease may actually require a different drug target.
Sure.
And this process, we think of it as even a one-time shot, but really, as a person changes over time, the immune system responds, you may actually want to be shifting drugs and so forth. So there's a whole information processing that can, a human immune monitoring piece that can come and affect diagnostics of which drug you would want to be using. So all of these are essentially offerings that CytoReason provides drug companies as they're thinking about a drug, rather than making this a trial and error based process. We want to be switching to a model based process in which you're using all the data that we have in one unified model to ask these questions.
And it's, looking at sort of that, well, say a service offer. I mean, you for a company that was, only started in 2016, you have obviously excited many, you have deals with Pfizer and GSK and Summit Pharma in Japan and the Parker Institute. So you, in a very short period, I don't always see sort of small biotech companies sort of get into pharma so quickly and get so many deals done. But it clearly, you're definitely sparking the interest of organizations, which, thinking, I used to be at GSK. We used to move very slowly. It seems like the uptake of some of these newer technologies at the beginning, wow, I guess we're in a decent era where they're beginning to understand this. Are you seeing it, once again, non-confidentially, is it more the interest in drug early discovery, or are you also seeing, like you were saying, retasking maybe current drugs, repurposing of current drugs for other purposes, or as you were mentioning, the modulation of the patient population in clinical trials? Are you sort of seeing the interest across some of these companies in applying CytoReason technology to all these different platforms?
Yes, we're seeing it not just in early discovery, but in fact, there's a strong interest throughout the life development of the life cycle of a drug. Mm-hmm. Actually, the stakes get higher the further along you are in the pipeline. There's obviously, once the drug is off patent and so forth, this becomes much more difficult, I think, for investment. But up until that point, there's actually quite a lot of interest in terms of both understanding the drug better, understanding what would be the next target to hit, given we now know that there are people who respond to the drug and people who don't, right? So, and certainly expanding the drug to other markets and indications.
Excellent.
And I guess just to answer one point, yes, I'm happy to say we've been successful so far. But I think this really speaks to the unmet need in the industry. People have been collecting this data for a long time. The genomics revolution is now 20 years old, right? I mean, the first microarrays and so forth came around in 96, 98. So people have been amassing data, knowing that it needs to be important. But the question is, are you able to leverage the data? And I think the industry has been feeling like it's been worthwhile. you can't not measure. They all recognize that the measurements are important, but there's been a disappointment of how do you actually get the bang for the buck from this. And this is really moving just from the measurements to what CytoReason is doing, is building this model that it incorporates many, many different data sets and allows you to kind of leverage this much more. It's always the case, I think, that analytics lags behind measurements, and the ability to inform is always, follows the ability to measure. So we always are excited about measuring, and then we discover it's difficult to actually interpret. Think about genome sequencing. You can sequence now cheaply, but you understand 5% of the genome, right? Isn't that disappointing that for the rest of the 95%? And this happens on every layer of biology. We measure so many genes, and yet about 16% of the genes cover 80% of what we understand about we annotate the information for. So the majority is still not known.
Very fascinating. Fascinating. How is everything, just switching now to sort of the general environment ecosystem? You're in Israel, you're an Israeli-based company. What is in 2019, how is the Israeli sort of biotech venture scene looking right now? I mean, I've noticed a lot of U.S. venture funds that have set up Israeli units. Obviously, it's always been a wonderful area for pumping out new discoveries. Is it a comfortable ecosystem? You see things growing. Just sort of a top line on what's happening in Israel today on biotech.
So certainly I guess the biotech industry in Israel is growing quite strongly. It hasn't been that case for previous years. It's really been expanding. Also, I think Israel in particular has been strong in informatics for many years, high-tech industry, and particularly in bioinformatics. And so this understanding of taking biology and the informatics and pushing it together. There's quite a lot of companies from Israel who are developing informatics-based solutions for life science applications. And what has been nice is both at the governmental level and at the venture capital level, there's been understanding of this is Israel's next kind of revolution. Israel's done agriculture, security, high tech. So there's recognition of this at a kind of both at the governmental level. There's a genome sequencing project that's happening for a large population of Israel. There's a lot of money being poured into venture capital firms and so forth. So we're hoping for a bright future there.
Excellent. So, before we wrap up, we technically just go back to a few sort of personal questions. Wanted to ask you about any mentors, advisors, influencers along the path that, kept you in this, at a times where maybe experiments weren't going the right way, or you're disheartened or something like that really, said, you got to keep doing this. Something's going to come out of it. And if they weren't there, you would have gone in a totally different direction. Anyone special come to mind that you want to shout out to?
There's so many. I mean, almost, science is a difficult task, and certainly taking this road is never easy for any scientist. Everyone has doubts. Experiments always fail. But I guess, and so really, it's been quite a long road with a lot of people to thank. But maybe actually of relevance most recently has been Professor Mark Davis, who's a world-leading immunologist, and where I did my postdoc. And it was really this just having hours of conversations with him and understanding how big data, artificial intelligence can really play a role in changing how we think about the immune system. I think that's really been a strong effect on what I do today. So
wonderful.
Shout out to Mark.
Excellent. And one last thing before we wrap up, we typically give everybody on the show, we move from science to science fiction, but I have to ask this question. In my hypothetical time machine over here, I'm going to let you, Professor Shen-Orr, travel anywhere you want to go. You can go meet a scientist, a celebrity, an artist, a philosopher from any time. Is there somebody in history that you would have wanted to meet And if so, if you had that ability, what would you want to talk to that person about? You can take your time and think about that.
That's not an easy question.
Not at all.
You also need to answer non-trivially, because I could easily go back and Dr. Darwin.
Yeah, go to Darwin, that's fine.
Edward Jenner, who had the faith to vaccinate a kid with.
Oh yeah.
So yeah, difficult to choose. But yeah, I think actually, again, I'll stick with immunology. I think Edward Jenner is actually an interesting character to see. This is really the basic intuition of how the immune system works.
Mm-hmm.
Vaccination, and also really a lot of what we end up striving to do right now is immunotherapy. It's a very basic understanding, and just looking at the history of immunology, I think is a fascinating history. And yet, as I'm answering, I guess true to the interdisciplinary nature of what I do, Alan Turing would be my my second guy.
We've had some touring.
Because it really is this, right? You need to be merging them both. So.
Absolutely. No, perfect choices. Well, I really want to once again thank you again so much. I mean, I have a lot going on. Really appreciate the time that you spend with us today and everything you're doing. And it is truly while people point out, the brain, the heart, the immune system, I completely agree with you, is one of those sort of untapped areas of so many different potential unmet medical needs. And it just really, thank you for everything you do working there. For everybody, once again, watching the show, Professor Shai Shen-Orr, Associate Professor at Technion, Israel Institute of Technology, and founder and Chief Scientist of the CytoReason company. Thanks so much for joining us today. Really, very much appreciate your time.
Thank you, Ira. It's been great. Thank you so much.
It was a pleasure.